we're talking about cellular reproduction, right? How can we actually produce new copies of individuals, basically making new individuals? And the mechanism that we hit upon was normal, regular cell division, which was mitosis, right? And remember, the key thing about mitosis is that absent mutations, we end up with individual cells, right, that got split apart during cytokinesis, right, that are genetically identical to the parent cell. That's the key thing about mitosis. You get genetically completely identical daughter cells, except for any mutation that may have happened during the S phase, right, during synthesis, during the DNA replication, right, prior to mitosis. Okay, so what might happen? So let's assume that we are starting here, or we're following a population of bacteria, right? Bacteria, their mode of reproduction is just that, is mitosis, right? It's simply splitting of cells from one parent cell and then through mitosis producing two daughter cells, right? So let's take a look what happens. Okay, so let's assume this individual reproduces and we have two cells, and then those two cells grow a little bit, and then they go through mitosis and produce two more individuals from them, and so forth and so on, right? So as we're going from one generation to the next, we're obviously increasing in the number of individuals. Now let's assume, which are all genetically identical, right? Now let's assume that at some point, one of these cell divisions resulted in a mutation. Now, as we said last time, most mutations are very, very likely not going to be very helpful, right? They tend to be bad mutations, or they tend to produce a protein that is non-functional in most cases. But every now and then, you do get a mutation where amino acid gets substituted, for example, that actually is able to improve the function, for example, of the active site of a particular enzyme, right? So the enzyme now has a improved catalytic function, perhaps, or perhaps at least at that particular temperature, or whatever it happens to be, right? This is great, right? So this individual is now going to do better. So woohoo, we have new genetic variation that can now be acted upon through natural selection, and perhaps in this case it benefited that individual, so maybe that individual can go ahead and be more prosperous than its neighbors, right? Now let's assume that a different mutation happened in a different individual. So here let's assume that this mutation produces a new kind of protein, or a new allele of that protein, in this case maybe the yellow one, right? So it's a different one that happened in a different strain. And let's assume that this too happens to be a beneficial mutation. Beneficial mutations are really, really rare, right? So it's not as common as this, obviously. So these mutations are going to be really, really far apart. Now, wouldn't it be fantastic if you could be an individual that I just put down here on the side there, if you happen to be an individual that could have both the blue beneficial mutation and the green beneficial mutation? That would be absolutely wonderful, right? So the question that I have for you is, how can we get there? How can we have an individual that comes across both beneficial mutations? So if mitosis is all that we have at our disposal, the only way that we can produce a bacterium like this one down here that has both the blue and the yellow beneficial mutation is if along that line first a mutation happened that produced the blue, and in that same line it just so happens that the other yellow mutation happened. Because now, mutations happen all the time, but as we said, beneficial mutations happen really rarely. So to be lucky enough in that individual to first have that one mutation, and then that other beneficial mutation just by chance happens to pop up during S phase of some subsequent mutational step, it can happen, but it would probably take a very long time for that to actually occur. Right, so you can see that this is, if all that we have at our disposal is mitosis, it is a really ineffective step to accumulate alleles that are beneficial for that particular organism, right? You probably came across the other solution. It's a duh solution, right? What we can do, if only we can find a mechanism for this, we could say, oh, well, why don't they share their mutations? The one came across one, the other one. Well, why don't we just swap them and we both benefit? And the process that does this is obviously fertilization, which is just bringing together two individual cells and fusing them into one resulting cell, right? Now there's an issue with that as well. Imagine this were to happen, where you just have two parent cells, and they merge their entire genome with one another, and then you continue doing this process over and over and over again. What kind of issue do you think you will run into doing that? If we want to have a fertilization at some point where we merge two cells, we have to get to a point where we turn a diploid cell into a haploid cell. And as we said, the cell division that does this is called meiosis. So our next goal now is to try to understand how that actually happens, how we can actually go from that diploid cell to a haploid cell. Meiosis starts just like any other mitotic cell division, which is the very first thing before we even enter meiosis, is that we go through an S phase where we replicate our DNA. This may seem a little bit confusing because the whole point of meiosis is simply to reduce the chromosome number, and it just happens to be one of those happenstances where life got stuck with doing it a particular way. So the cell that is getting ready for meiosis consists of a diploid cell that has chromosomes that came from that individual's father and mother, right? Two homologs for each one of the chromosomes, and each one of these homologous chromosomes is a replicated chromosome that consists of two sister chromatids. The final stage that we want to end up in is gametes, be it egg cells or be it sperm cells, that are haploid, that only have one of each homologous chromosome, and that only consist of a single chromatid, where each chromosome is just one molecule of DNA, non-replicated, right? So we have to accomplish two things. We have to separate the homologous chromosomes into different cells in order to go from a diploid to a haploid cell. And secondly, we have to then split the sister chromatids of each one of those chromosomes up again into separate cells in order to arrive at our haploid cell, where every chromosome just consists of a single molecule of DNA, a single chromatid, as it were, right? Where each chromosome is the same as a single chromatid. And it's exactly in that sequence that meiosis actually does it. And as you can tell, what this requires is actually two cycles of cell division, right? So we split meiosis into meiosis 1, which is the first cell division, and meiosis 2, which is the second.
So in this unit, one of our main goals is to understand how new genetic diversity arises. And so far we've talked about 4 mechanisms that generate genetic diversity. The first one we've talked about is mutation. And again, mutation happens in DNA replication. So any organism that replicates its DNA is going to experience mutation. And that's all life on Earth, because all life on Earth has DNA, and all life on Earth copies its DNA when it's reproducing. But what we're going to look at now are 3 mechanisms that produce genetic diversity that are exclusive to those organisms that reproduce sexually. And those are independent assortment, recombination, and fertilization. And we'll see that 2 of these, independent assortment and recombination, arise during the process of meiosis. While organisms are making gametes, new combinations of chromosomes and alleles can form during the process of meiosis. And the last one, of course, is fertilization. That happens when 2 of the gametes that are produced in meiosis will come together to produce a new offspring. And so what we're going to look at first is independent assortment and recombination. And so we're going to use what we've learned about meiosis to understand how these processes work and how they generate genetic diversity. So we're going to consider a diploid organism that has 3 different chromosome types. And because it's a diploid, it's going to have 2 homologs of each chromosome type. And so that means it's going to have 6 total chromosomes. Of course, this diploid organism first formed when 2 gametes, 2 haploid gametes, came together and fertilized each other to form a zygote. And so what we're going to look at first is the 2 gametes that formed our diploid organism. So we can start with the maternal gamete, the egg cell. And the egg cell, of course, is going to be a haploid. It's only going to have 3 chromosomes, 1 of each chromosome type. Of course, the sperm cell will be just the same. It'll have 3 chromosomes, each of a different chromosome type. And of course, when we bring these 2 together, when we fertilize the egg with the sperm cell, the genetic complements will combine and we'll end up with our 6 total chromosomes. The first thing we're going to look at is independent assortment. We want to see how independent assortment generates genetic diversity. To understand that, we have to walk through the process of meiosis to watch how the gametes form. So in the first step of meiosis, the homologs of each chromosome type align side by side at the center of the cell. And then in the first cell division of meiosis, meiosis 1, we separate the homologs. We divide those two homologs up from one another and place them into separate cells. Now in the example that we've shown here, it was all the maternal chromosomes that aligned on the left-hand side, and it was all the paternal chromosomes that aligned on the right-hand side. And so when we separated these homologs into separate cells, you can see we ended up with one cell that had nothing but the maternal chromosomes, and the other cell that had nothing but the paternal chromosomes. It turns out this is not the only way in which the chromosomes can align themselves. Which chromosome ends up on the left and which ends up on the right is entirely random. The important thing for independent assortment here is that the alignment of one chromosome type is entirely independent of the alignment of the next chromosome type. What that means is, when you have multiple chromosome types, you can end up with many different combinations, many different possible alignments of these homologs. And so when they're split into separate cells, during meiosis 1, you can have many different combinations of maternal and paternal homologs in those daughter cells. Perhaps the most succinct way to say this is that different chromosome types will assort independently. So we've seen how independent assortment can create different combinations of homologs in the daughter cells in meiosis. But in this class, we're not really concerned about homologs. What we're more concerned about are traits of organisms and the alleles that cause those traits. As we'll see, because the genes that control traits are on those homologs, that this process of meiosis and this process of independent assortment can yield new combinations of alleles and the traits that they convey. So we can have entirely new combinations of traits in organisms due simply to independent assortment, due simply to the normal process of meiosis. And so what we're going to show here is an example in which we're looking at two different chromosome types. And in particular, we're looking at two different genes. Each one of these genes is located on a different chromosome type. So for example, let's say that there's a gene for eye color, and that's located on chromosome type number 1. And there's a second different gene that controls hair color, and that's going to be on chromosome type number 2. Now in this example, we can see that there are different alleles of these genes on the different homologs. So for example, the mother of this diploid individual contributed an allele for brown eyes, and the father contributed an allele for green eyes. And again, those alleles can be found on the chromosome type number 1. If we're looking at the gene for hair color, we can see that the mother contributed the alleles for black hair, and the father contributed the allele for red hair. As we saw during meiosis 1, when the homologs are aligning at the center of the cell, again, they do so randomly, and the different chromosome types will do so independently of one another. And so for just these two chromosome types, we can see that there's two possible arrangements of the homologs at the center of the cell. So independent assortment only applies to two different genes that are on different chromosome types. Independent assortment cannot generate new combinations of alleles if there are two genes on the same chromosome type. So is it possible to get genetic diversity for two genes that are on the same chromosome type? The answer is yes, through the process of recombination. But to understand how recombination works, we're going to take, again, a closer look at what happens during the process of meiosis. Now, up to this point, we've said that one of the first steps in meiosis is that the homologs of each chromosome type will align side by side at the center of the cell. And it turns out they come a lot closer than that. They actually come in physical contact with one another and form what we call tetrads. And tetrads are nothing more than a pair of homologous chromosomes that have come together in physical contact in the middle. So during the process of meiosis, after these tetrads have formed, after these homologs have come together to form a tetrad, we can have what's called crossing over. A small segment of one homolog will cross over the same segment of the other homolog. In other words, two non-sister chromatids will cross over between these two homologs. And when this happens, the crossed over segments can actually break off and become part of the other homolog. So segments of non-sister chromatids will swap from one homolog to the other. We've essentially exchanged chunks of the chromosomes. So during the process of meiosis, crossing over happens at least once per pair of homologs and can happen several times along the length of the chromosome. So through crossing over, we end up with chromosomes that are composed of different segments of the maternal and paternal homologs. But again, this class, we're not focused on the homologs themselves, but we're interested in the alleles and the traits that those alleles convey. So we're going to look at an example of how crossing over can generate new genetic variation. Again, as before, we're going to look at two different genes. We're going to call one gene A, and it has two alleles, allele 1 and 2. And we're going to look at gene B, which also has two alleles. So we can see from this diagram that both genes are located on the same chromosome type. They're both on the same physical molecule. We're going to say for this example that the maternal homolog has the black allele for gene A and the brown allele for gene B. And we'll say that the paternal homolog has the red allele for gene A and the green allele for gene B. Again, before meiosis begins, our chromosomes are replicated. In the first step of meiosis, our replicated homologs will come together and form those tetrads. They'll come into physical contact with one another. And it's at this point where the crossing over can happen between our non-sister chromatids. And you can see that if the crossing over happened at some place along the chromosome that was between our gene A and gene B here, we could end up with new combinations of alleles on our chromatids. So you can see that two of these are identical to the chromosomes that were in the original diploid organism. We can also see that the two in the middle have new combinations of alleles. And you can see that these new combinations would not have been possible without the process of crossing over. To summarize, the process of crossing over causes recombination. That is to say, crossing over refers to the physical exchange of molecules, one molecule overlapping the other, exchanging bits of the non-sister chromatids between homologs. But that physical process causes new combinations of alleles to form, and that's what recombination is. So crossing over, the physical process, causes recombination of alleles. So both the processes of independent assortment and recombination take place during the overall process of meiosis. So as we're generating gametes, independent assortment and recombination act to shuffle alleles around and generate new combinations of alleles in the gametes. The fourth mechanism of generating genetic diversity we're going to talk about is simply fertilization. Again, fertilization is nothing more than fusing two of these gametes together. And when that happens, their individual haploid genetic complements come together to make a diploid individual in this example. And as you can imagine, bringing in genetic material from two entirely different organisms can generate genetic diversity on its own. We end up with new combinations of alleles that were present in these two different organisms united into one organism. And so we might have a combination of alleles that's never been seen in any organism ever.